I feel like usually when we talk about getting a medical diagnosis, there is this expectation of just pure mechanical precision.
Oh, absolutely.
Like, you break your arm, the x-ray shows that jagged white line, and the doctor just points at it and says, there it is, problem found.
Right, and it's comforting,
Yeah.
because it's binary. The evidence is just staring right back at you on a screen.
Exactly. But then you step into the world of oncology and deep tissue analysis, and suddenly that metaphorical x-ray machine is just, it's broken.
Oh, completely broken.
You're left staring at this, $5,000 spatial transcriptomics map of a tumor, a map that is supposed to show you exactly which cancer genes are firing and in what specific physical locations.
And instead, massive chunks of it are simply blank.
Right, just undetected, missing data. So today, for our deep dive, we are looking at a new AI model called DeepSpaCE. And this AI looks at cheap, black and white, basically structural sketches of tissue, and it perfectly hallucinates that missing million-dollar genetic data.
Hallucinates it, yes, but crucially, it gets it right, which is wild.
It is.
We are looking at a profound shift here for the listener to wrap their head around. We're moving from relying entirely on physical chemical reactions in a wet lab to trusting algorithmic predictions.
Right.
And in cancer research, understanding not just what genes are active, but exactly where they are active in the tissue. I mean, that is literally the difference between catching a tumor's microscopic invasion and completely missing it.
Okay, let's unpack this, because we really need to set the baseline here. The contrast between these two technologies is just staggering. So on one hand, we have the cheap street sketch. What are hospitals actually using as their day-to-day tissue map right now?
Well, the absolute bedrock of pathology is H&E staining. It stands for hematoxylin and eosin.
Okay.
Take a wafer-thin slice of tissue, you put it on a glass slide, and you treat it with these two very old, very cheap dyes, and you get this beautiful, high-contrast visual map.
Right, it's the pink and purple one, right?
Exactly. The hematoxylin turns the cell nuclei, this dark purplish blue color, and then the eosin turns the cytoplasm around it a bright pink. So it gives pathologists a really clear view of the cell structures, the shapes, the tissue architecture.
And this is the key. That's all it is, right? It's just structure.
Right. It doesn't tell you a single thing about gene expression.
Okay, so it's basically just showing us the physical layout of the buildings in a city, but it tells us nothing about what's actually happening inside those buildings.
That is a perfect analogy, yeah.
So to get the genetic activity, the actual molecular functions mapped to those physical locations, you need spatial transcriptomics. And the Nature Scientific Reports paper we are pulling from today uses the 10x Genomics Visium platform for this.
Right, which is highly advanced.
Yeah, so basically you place the tissue on this specialized slide that has a grid of microscopic spots. And each spot is loaded with these little barcoded tags. So the RNA from the cells binds to the tags, you sequence it, and the barcode tells you exactly where on the grid that specific genetic activity happened.
Right, and you end up with a map of thousands of spots correlating all that genetic activity directly onto the physical geography of the tissue slide.
But here's where I get stuck, and I think this is the core of the issue. If the Visium platform gives us this incredible, highly targeted genetic map, why do we need a complex AI model to step in? I mean, it sounds like we already solved the problem.
Well, you would think so, but the physical reality of a biology lab is incredibly messy.
Okay.
First, there's the cost. You're looking at thousands of dollars per single slide before you even factor in the sequencing costs. But the bigger issue, the one this paper focuses on, is physical failure. The paper highlights a massive flaw called permeabilization errors.
Permeabilization errors, meaning the chemicals literally fail to penetrate the tissue properly.
Exactly. To get the RNA out of the cells and onto those barcodes you mentioned, you have to apply chemical reagents to break the cell membranes open.
Right.
But human tissue is highly variable. Imagine you are trying to soak a dense sponge.
Oh, a sponge.
But certain patches of the sponge have, I don't know, a slightly different density or maybe a higher fat content. So those specific patches just repel the water and stay bone dry.
Oh, wow. So the wash just bounces off?
Exactly. In the lab, if a region of tissue doesn't permeabilize correctly, no RNA is released, period. So on your final digital map, that entire physical area just registers as a void, a blank spot.
And the paper actually shows a terrifying example of this. I was looking at the figures, and they had a stack of consecutive slices of human breast cancer tissue.
The D slices, right?
Yeah, literally slices taken right next to each other from the same tumor, labeled sections D1, D2, and D3. And in D1 and D3, the entire right bottom region of the map was entirely blank.
Completely devoid of data.
And then in the middle slice, D2, the upper right region suffered the exact same fate, just empty space on a $5,000 test.
Which is a diagnostic nightmare, you know?
Yeah.
You have a known breast cancer tumor, and a massive chunk of your high-resolution data is just missing, simply because a microscopic chemical wash didn't soak in evenly.
So, okay, let me do a quick recap here to make sure we are all tracking this.
Sure.
You've got this cheap structural pink and purple H&E map that works every single time. And then you have this highly expensive, deeply informative genetic map that is currently failing us because the chemicals won't soak in properly.
That is the situation.
Which leaves you wondering, and I want the listener to picture this. Imagine you were a pathologist looking at a blank spot on a $5,000 slide. How do you actually know if the tissue in that spot is just dead and inactive, or if your chemicals simply failed you?
You don't. You really don't. And that is exactly the void that DeepSpaCE was designed to fill.
DeepSpaCE.
Yeah, which stands for Deep Learning Model for Spatial Gene Clusters and Expression. It is built to look at that cheap H&E image, the pink and purple one, and mathematically predict what the genetic expression should be in those blank spots.
And they built this using a convolutional neural network, right?
Yes.
Specifically a VGG16 architecture, which I guess makes sense given its depth and its reputation for extracting highly complex image patterns.
It's very good at visual feature extraction.
Let's try to visualize how this actually works for people. Think of the CNN as an expert meteorologist looking at a black and white satellite photo of the Earth.
Okay, I like that.
Right. They can't literally see the temperature or the actual raindrops in the photo, but by analyzing the specific swirl, the density, and the texture of the clouds, they can accurately predict exactly where a severe thunderstorm is happening underneath.
Right.
So in this case, DeepSpaCE is looking at the specific shapes, densities, and patterns of those pink and purple cells and predicting the hidden genetic storms happening inside them.
It's a really great way to think about it. The AI isn't actually reading the genes, it's reading the structural consequences of those genes.
But wait, if the AI is just guessing the gene expression based on the physical shape of the cells, isn't that just making a massive leap of faith? I mean, it sounds like it's stereotyping the tissue, like, oh, these cells look a little clumped together, it must be cancer.
It absolutely sounds like stereotyping. And honestly, that is exactly why the training methodology in the paper had to be absolutely bulletproof.
Right.
They used something called five-fold cross-validation. They didn't just let the AI guess wildly. They took the spot-level data from the breast cancer tissue, and they used 80% of it as a training set.
Okay, 80%.
Yeah, showing the AI both the H&E image and the true known genetic data at the exact same time, and they forced it to learn the actual correlations.
So it's building the rule book.
Exactly. Then they test it on the hidden 20% to see if its rules held up against reality. And they specifically focused on predicting crucial cancer markers, like ESR1, which is estrogen receptor 1.
And here's where it gets really interesting. In that middle tissue slice we talked about, section D2, remember that upper right region that was completely blank from the chemical error?
Massive void, yeah.
DeepSpaCE analyzed the H&E pixels in that blank area and boldly predicted that there was extremely high ESR1 expression there. The AI basically said, hey, I know the chemical test failed here, but based on the structural cloud formations I see, this area is flooded with estrogen receptors.
But an algorithm's prediction means absolutely nothing in oncology without physical validation.
Obviously.
To prove the AI wasn't just generating noise, the researchers took the adjacent physical slice of tissue and performed immunohistochemistry, or IHC.
Right, and IHC is the physical lab test where you apply specific antibodies to the tissue that only bind to a target protein.
Right.
And when they bind, they trigger a chemical reaction that literally stains the tissue brown right exactly where the protein exists. It's physical, undeniable proof. You can look at it under a microscope.
And when they compared the physical brown IHC stain to the AI's digital prediction, they matched almost perfectly. DeepSpaCE achieved a Pearson correlation coefficient of 0.600 in this validation.
Which, I mean, for anyone who hasn't spent time in a wet lab, a 60% correlation in the chaotic, incredibly messy world of biology is massive.
It is incredibly strong proof, It essentially confirms that DeepSpace can accurately see the genetic reality that the $5,000 chemical test missed entirely.
Okay, let's pause and synthesize this really quick.
Good idea.
We have an AI model acting as a meteorologist, looking at the structural clouds in sheep tissue images to predict the hidden genetic rain. It successfully hallucinates the missing data caused by lab errors, and those predictions are physically proven correct by real-world protein staining. That is a massive leap forward, but it also leaves you with this lingering thought, and I want you to think about this. If an AI can look at the physical shapes of cells and accurately map the genetic expression that modern chemical tests just miss, what does that mean for the millions of old, archived H&E slides currently sitting in hospital basements around the world?
Well, it transforms them from dead storage into absolute gold mines of unmapped genetic data.
Wow.
But, fixing errors on a slide is really just the baseline of what this model can do. What elevates this paper is how they use the AI to discover biological boundaries that were previously completely invisible.
That's the perfect pivot, actually, because the SPARC gene analysis in this paper completely blew my mind.
What's fascinating here is the specific mechanism of SPARC itself. SPARC, which stands for secreted protein acidic and cysteine rich, is a notorious invasion marker in cancer.
Okay.
It's a glycoprotein secreted by cancer cells into the extracellular matrix. Think of SPARC as a biological bulldozer.
A bulldozer, okay.
It actively alters the surrounding environment. It breaks down the normal structural scaffolding so that the cancer cells can physically migrate and invade healthy tissue. So high SPARC expression is heavily linked to metastasis and incredibly poor prognosis.
And the paper links SPARC directly to EMT, right? Epithelial mesenchymal transition.
Yes. So imagine a brick tightly mortared into a wall.
Okay.
That's an epithelial cell.
Yeah.
It is rigidly attached to its neighbors. EMT is the biological process where that brick essentially transforms into a moving vehicle, a mesenchymal cell.
Wow.
It loses its rigid attachments, it changes its physical shape, and it gains the ability to move. And when the researchers ran pathway analyses using the MSigDB database, they found that genes related to this EMT process were actually the most accurately predicted genes by DeepSpace.
Because the AI is looking at structure.
Exactly.
So, of course, it excels at spotting cells that are actively changing their physical shape to invade. It's looking for those bricks turning into cars.
Precisely. It is exquisitely tuned to recognize the visual signatures of structural transformation.
Which leads to this incredible visualization contrast in the paper. When you look at the raw original spatial transcriptomics data for SPARC expression, it was measured across roughly 2,100 individual spots on the grid.
Right.
And it just looked like a smattering of blurry dots. The resolution was just too sparse. You could not actually see the boundary where the tumor was invading the healthy tissue.
It's like looking at an 8-bit video game when you really need to see a high-definition photograph. The resolution is just too low to make critical clinical decisions.
But then they use DeepSpace to perform what they call super resolution. The AI looked at the blank spaces between the 2,100 measured spots, and it mathematically predicted the gene expression for all the unmeasured gaps. It multiplied the data points to over 6,600 spots, and suddenly the image just snaps into 4K focus. A stark, clear boundary of tumor invasion appeared, flawlessly mapping the SPARC expression to the visual histology of the cancer actively pushing into the microenvironment.
It revealed a highly functional biological boundary that simply did not exist in the raw data. Right. And we should note, they pitted DeepSpace against a rival generative AI model called XFuse to see if this was just a fluke.
Oh, right. I wanted to ask about that.
Yeah, and XFuse completely collapsed on this task.
Why did it fail so badly, though, if they are both AI models trying to do the exact same thing?
It comes down to real-world optimization. So the raw Visium slides have these tiny alignment dots around the edges. They're called fiducial frames.
Okay, fiducial frames.
The scanning software uses them to orient the image. XFuse's algorithm couldn't distinguish those alignment dots from the actual biological tissue, and it totally choked when it hit the permeabilization errors.
It got confused by the borders.
Yeah. But DeepSpace was custom built for the messy reality of these specific datasets. It ignored the alignment frames, and it effortlessly bridged the missing gaps.
So to bring this all together again, we took this low-res, blurry map of an invading cancer bulldozer, which is a SPARC, and we used DeepSpace to multiply the data points, snapping the invasion boundary into perfect focus.
Yeah.
And it utterly crushed rival models because it actually understands the messy realities of the lab equipment. It's a brilliantly designed piece of software.
Really is.
But it prompts a somewhat chilling question for anyone working in oncology. How many critical tumor boundaries are we currently missing in hospitals right now, simply because our standard spatial grids aren't dense enough to show us the full picture?
That's a sobering reality. But the researchers didn't stop at super resolution on a single slide. They decided to push the model to its absolute mathematical limits to see if it could imagine entirely unmeasured physical spaces.
So what does this all mean for the future of diagnosis? We move to the most ambitious application in the study, which is tissue section imputation.
Right.
They didn't just want to fill in gaps on one slide. They wanted to predict the genetic map of an entirely unmeasured middle slice of tissue. Remember our stack of consecutive slices? D1, D2, and D3.
The breast cancer slices.
Yeah. They tasked the AI with predicting the complete genetic expression of section D2, using only the data from the slices above and below it, D1 and D3.
And to achieve that, they used semi-supervised learning. Specifically, a teacher-student loop method known as the noisy student.
The noisy student, I love that name.
It's great. Let's break down how this mechanism actually functions, because honestly, it is counterintuitive.
Please do.
First, you build a teacher model. You train this AI purely on the hard, known genetic data from slices D1 and D3.
Okay, so the teacher memorizes the textbook for the top and bottom slices.
Right. Then you hand the teacher model a bunch of entirely unlabeled H&E images. These were other slices from the tumor, labeled sections A, B, and C.
Okay.
The teacher predicts the gene expression for these unknown slices, creating what we call proxy labels. Essentially, they're highly educated guesses. Finally, you create a brand new AI, Student 1. You train Student 1 on both the real hard data from D1 and D3 and the proxy-labeled guesses from A, B, and C.
Okay, I have to challenge this because logically it just feels backwards.
I know it does.
Why would training a new AI on the guesses of an older AI make it smarter? Aren't you just reinforcing the teacher's mistakes in the new student?
You would completely think so. But the proxy labels actually act like practice tests. If a student only studies the official textbook, the known D1 and D3 slices, they just memorize the specific layout of those two individual slides. They overfit to the data.
Oh, okay.
By feeding the student model unlabeled slices with educated guesses, the AI is forced to look at brand new cell architectures, different structures, and slightly different tumor environments. It forces the neural network to figure out the underlying visual rules of the tissue rather than just memorizing the map.
That makes so much sense.
Right. And they iterated this. Student 1 becomes the new teacher, generates better practice tests, and trains Student 2. And they pushed this process all the way to Student 5.
And the statistical payoff is undeniable. If we look at SPARC again, the correlation accuracy in the original teacher model hovered around 50.9%. But by the time it reached the Student 4 model, that accuracy jumped to over 61%.
Which is huge.
In bioinformatics, a 10% leap achieved entirely by having an AI practice against its own generated tests is massive. DeepSpaCE totally shattered the baseline statistical models, which were just trying to guess expression based on the average brightness or the RGB color values of the pixels. DeepSpaCE is extracting architectural rules, not just reading colors.
This raises an important question, though. The reality check. Does this magical noisy student work on any image you feed it?
That is the million-dollar question.
The researchers were incredibly rigorous here. They ran negative controls to test the absolute boundaries of the model. They tried training the student models by feeding them random data, permuted data. They even fed the AI thousands of irrelevant images of dogs and cats from the ImageNet database.
Wait, really? They fed pictures of cats to a breast cancer predicting AI?
They had to. It's a control to prove that the AI actually needs biologically relevant structures to generalize its rules. And predictably, the cat pictures made the accuracy absolutely plummet.
Right, makes sense.
But here's the critical part. They also fed the AI images of real breast cancer H&E slides, obtained from a totally different massive database called the Cancer Genome Atlas, or TCGA.
Okay.
And the accuracy plummeted again.
Hold on, why? I mean, if it's real breast cancer tissue, shouldn't it help the AI learn the structural rules even better?
If we connect this to the bigger picture, it highlights a massive hurdle in medical AI called domain shift.
Domain shift.
Every pathology lab in the world prepares their slides slightly differently. They use different brands of scanners, slightly different chemical dye mixtures, different lighting setups, different slide thicknesses. The AI is hypersensitive to these specific batch protocols.
Oh, wow.
DeepSpaCE is a localized genius. It becomes an absolute master at understanding the specific visual dialect of a single lab's equipment. But the moment you show it a slide from a hospital across the country, that slight shift in color saturation and texture completely confuses it.
Okay, so let's summarize this final piece of the puzzle. We've got this teacher-student loop pushing accuracy through the roof by taking practice tests against unlabeled data. It can literally hallucinate a missing slice of the tumor with incredible accuracy.
Yeah.
But it is geographically locked, it's highly sensitive to one lab-specific scanner, and dies, which presents a massive hurdle for you, the listener, to consider. If an AI requires highly specific lab-tailored data to achieve this level of genius, how do we ever standardize this technology so that a clinic in rural Nebraska gets the same predictive power as a massive research hospital in Boston?
It is the defining challenge for the next decade of biomedical AI. How do we build models that are robust enough to ignore the scanner brand, but sensitive enough to catch a microscopic cancer invasion?
So, to pull all of this together, DeepSpaCE is not just a clever algorithm patching holes in a dataset. It represents a fundamental shift in how we interrogate biology. It proves that we can derive hidden histological characteristics, track tumor invasion boundaries, and generate incredibly detailed 3D biological interpretations from a single, flawed, highly expensive dataset without spending another dime on physical lab experiments.
And its value is permanent. I mean, even as physical spatial technology improves and those physical grids get denser, the sequencing costs to analyze those millions of new spots will rise exponentially.
Right, the money factor.
The ability of an AI to cleanly impute and predict missing sections will remain perpetually valuable. It bridges the gap between our physical limitations and our need for complete biological data.
It truly forces a shift in perspective. For a century, we have relied entirely on physical reality to diagnose disease, the chemical reactions, the physical stains, the actual binding of molecules. But as algorithms like DeepSpaCE become capable of perfectly predicting the invisible molecular landscape from a simple pink and purple sketch, where does the ground truth actually live? Is it in the expensive chemical reagents that sometimes fail to soak into the tissue? Or is it in the hidden pixel patterns that only an algorithm can see?
It's a philosophical question disguised as a technological one.
Thank you for joining us on this deep dive. Next time you see a medical image, remember there is always more hiding in the pixels. You just have to know how to look.